A cell is always changing, and these changes are difficult to keep track of. One method used is to study the interactome. The suffix "ome" is used to denote a sense of wholeness or completeness and is used in many ways, such as in genome or, as we see today, interactome. From this explanation, we can see that interactome refers to the complete set of physical interactions within a cell, and the molecules involved in these interactions can be proteins, genes, individual nucleic acids, and other small molecules. An interactome can be used on different scales. We may look at the entire set of molecular interactions of a cell, or a set of interacting molecules in a particular pathway. For example, the mitogen-activated protein kinase pathway, also known as the MAPK pathway, is essential to cell communication and function, and it can be viewed as an interactome, thus creating an image of the exchanges that occur between the molecules. When we look at the MAPK pathway in class or in a textbook, it is often depicted as something like this. However, in reality, the process is more complex, and there are multiple interactions between all of the components involved. There are a variety of interactomes within a cell. Frequently discussed interactomes include gene regulatory networks, metabolic networks, gene disease networks, virus host networks, gene interaction networks, protein-protein interaction networks, and many more. The interactome is important to researchers across many disciplines. But why exactly is that? Well, the processes that occur in a cell can be very intricate and linked together in a variety of ways. These interactions, if not optimal, can result in disorders. Recognizing this allows these interactomes to become targets for drugs or therapies. There's an abundance of research on different interactomes, including those of well-known diseases. One such example is schizophrenia, for which interactome-based studies have been used in the investigation of the disease in order to find specific gene associations resulting in its pathology. Here, we can see a visual of the schizophrenia protein-protein interaction network. Genes associated with schizophrenia are shown as dark blue nodes, while the known indicators are shown as blue nodes and novel indicators as red nodes. How exactly can we analyze an interactome? Well, interactomes can be analyzed with many different computational methods. Currently, a popular model for studying interactomes uses the yeast-2 hybrid model. This technique uses a reporter gene, which encodes a protein receptor or enzyme that can then bind an imaging probe to allow for visibility. This technique can be used to validate a predicted interaction, as it allows the reporter gene to be translated and transcribed only when two proteins of interest interact. As a result, the imaging probe will allow us to confirm or reject the interaction of two proteins. Another method used is homology transfer. This is a process during which researchers predict an interactome in a species using data from another species with which it shares a common ancestor. This method may present some flaws, as interactomes can change throughout evolution, and despite there being homology between two species, this does not indicate an exact similarity. Therefore, the interactomes may end up significantly different. Finally, another approach used by scientists to analyze interactomes involves using algorithms based on experimental data and digitally created atomic models of protein-protein interactions. These analysis strategies present problems, such as high false positive rates, essentially predicting theoretical interactions where they might not happen in reality. Rather than looking at components of a cell as unrelated parts, conceptualizing it as a complex web of interconnected molecular structures is useful. Interactomes can be analyzed on a large scale, looking at the interactions between viral and host proteins, for example, or on a smaller scale, such as interactions between individual nucleic acids. These interactomes are important for studying how the pathways that lead to various diseases interact, which allows researchers to identify targets for drugs and therapies. The use of the yeast-2 hybrid model, homology transfer, algorithms, and digital models allow for the study of these networks. Interactomes summarize complex processes into webs of molecular interactions, and we just summarized interactomes, creating the world's first Interact TMM.